that is NBRI Lucknow. He is heading Plant Diversity, Systematics and Herbarium Division of the Institute. Dr. Nayaka has about 24 years of experience in the area of lichenology. His research interest includes lichen taxonomy, biogeography, and bio-prospection. As a lichen taxonomist, he has so far discovered 40 species as new to science and reported over 134 species as new to India. Dr. Nayaka has authored 3 books and about 200 research papers in peer-reviewed journals. He has successfully handled a total of 35 research projects as team member, then PI, co-PI, or coordinator. So far, he has guided 5 students for PhD and at present guiding 8 scholars. He is a fellow of 7 and life member of 8 academic associations. He is the founder member and secretary of Indian Lichenological Society. He has traveled extensively in India for exploration of lichens. He visited Antarctica twice for studying diversity of lichens in Shirmachar Oasis. Also, he had visited Armenia, South Africa, Thailand, Ukraine for attending meetings and conferences. For popularization of lichens in India, he has written several articles in regional languages, delivered invited talks, organized 3 national and international conferences and 14 workshops. So he has trained more than 60 scholars all over country for carrying out lichen research. So with this brief introduction, I request Dr. Sanjeeva Nayaka sir to present his talk, and just for your information, sir, this is a heterogeneous group consisting of teacher participants from botany, zoology, microbiology, biochemistry, food science and nutrition, biotechnology, and home science students are there, candidates are there. So over to you, sir.
Yeah, very good morning, Professor Rajkumar, for the elaborate introduction. Can you all see my slide?
Yes, sir. Visible.
Yes, sir.
Yes, sir.
Yes, seeing.
So, as introduced by Professor Rajkumar, I am working in the area of lichenology since 24 years now. So I work on various aspects. Basically, my interest is lichen taxonomy. So today we will be having a 1.5 hour marathon talk by me. So so I will try to cover the main aspects of the lichenology in very generalized manner, not in I will not put too much of scientific information. To make you all understand, I'll make it a much generalized talk today. So you can all switch off your mute your mic and also the videos. So so the topic for today's discussion is diversity of lichens and their bioprospection for human welfare. So first of all, let me tell you what are lichens. By this time, you all know what are lichens. At least you know that they are the symbiotic association between fungus and the algae. This you all know. So, but the lichens are not that simple, because if you bring one fungus and one algae together, although they start living together, we cannot call them as the lichens, because there should be some association, there should be some living together, interaction together. So what happens when the fungus and an algae come together and form the lichen thallus? They lose their original identity. That is the interesting part here. Fungus and the algal part lose their original identity, and then they start behaving like a single organism. So here, that morphological and physiological changes should happen here. Then only we call them as lichens, lichenization. Otherwise, they are not lichens. They're living together. So in the lichen thallus, you might have seen this kind of picture here, the last one here. So this is nothing but the lichen, what you are seeing from outside. So when you cut them, you will see the algal thallus. You can see in the first picture here, ball kind of structure, what you see here, that is the green algae, and the rope kind of structure, what you see, this is the fungal mycelia. So here in the second picture, you can see the algal layers, algal cells, ball kind of structure is attached here. So majority here is of the fungus, and algal portion is only so little. So 10% algae and 90% fungus. And the fungus is the one which is giving shape, structure, and color to the lichen. So it is otherwise that, fungus, they don't have chlorophyll, they have heterotrophs, they have to depend on the other organisms or dead organic matter for their nutrition. This all you know. Since they don't have chlorophyll, they cannot do the photosynthesis, and that is why they have to depend on the others for their nutrition, food. So some are saprophytes which grow on dead organic matter. Some fungus are parasites. They live, their life is dependent on the other organisms, and some are mutualist. For example, these mycorrhiza. Mycorrhiza, what is the mutualistic association between fungus and the plant roots, right? Like that, in fungus or in the lichen, the fungus has made an association with the algae. So here, the fungus is important. Fungus is main organism here. That is why we classify fungus, or we classify lichens under fungal kingdom. So algae has no taxonomic importance, at least here, while grouping them under the kingdom system. They have a little bit of role in family wise when you are classifying them. Otherwise, the fungus is the major organism which plays a role. So 1/5 of the fungal species are lichenized. So that is another one interesting thing. So within the fungal kingdom, there are fungus which are mycorrhiza, which are lichens, like that. There are free living fungi also with it. And see, lichens were existing on this earth since time immemorial. We don't know from where it is, when it is there. At least for the 415 million years since the lichens are there on this earth, because that is the very good fossil record we can give. So from 415 million years ago, the lichens are there on this world. People are using it, people know about it, but they're introduced to the scientific world by the term lichens by this fellow, Theophrastus. Theophrastus is called as the Greek father of botany. He coined the term lichen. So actually people sometimes pronounce it as lichen, but it should be lichen. Okay, so that is what the pronunciation of lichens. So, but this fellow, he used the term lichen for the superficial of epiphytic growth of fungus kind of organism on the olive tree. So he did not define what are lichens, how they look. But this lichen term became much popular, and the author later on, they started using the term, these lichens, but they included moss, bryophytes, and many other similar looking organisms under lichens only. Later on, gradually the lichens were defined. Now we know that lichens, what are lichens. And then, these people earlier thought lichens are made up of single organism. They never thought the lichens are made up of two organisms. Carl Linnaeus, he is called as father of taxonomy. Although he studied the 80 lichen species in his book Species Plantarum, so he thought lichens are single organism. And Eric Acarius is called as father of lichenology. He was a student of Carl Linnaeus. So he was assigned to work on the lichens, and he again studied the lichens and 3,300 species of lichens he described under 40 genera, and then he coined several terminologies, and he found a base for the lichen study in the world. So that is why he is called as the father of lichenology. But unfortunately, he also did not know lichens were made up of two organisms till his time. Then these two scientists, Anthony DeBerry and Simon Sweetner, they were working on lichens independently. So they both could see the green structure in the lichen thallus. Then they started doubting it for the. Then they started doubting it whether it is a lichens, whether it is algae. So the Anthony DeBerry confirmed that the green structure is algae only with his colleagues and the subordinates. But the Simon Sweetner encashed this discovery. He reported his findings in a conference in the Natural History Society annual meeting in Switzerland, Swiss. So it became popular. But Simon Sweetner, he was highly criticized. People were not ready to accept that lichens are made up of two organisms, because there was a doubt, or there was a fear that these lichenologists may lose their identity. So since if they call as the if they call lichens as a fungus, they may go with the mycologist, or if they say it has algae, and they may go with the phycologist. So lichen as a identical separate classification unit, they may lose that identity. That is why there was a fear. So that is why they criticized. But Simon Sweetner fought till its last period time, and finally he proved that lichens are made up of two organisms, and now it is accepted worldwide. So then it is not only two organisms. Gradually, there are several organisms are found closely associated with the lichens. This is a chronology you can see. Before 1867, like it was lichens were considered as single organism. Then the two organisms were discovered, fungus and the algae. Then the people also discovered bacteria growing over lichens. Then the people discovered other secondary algae growing with the lichens. Then they discovered protists, virus, yeast, so many things. They gradually started discovering the lichens. And this association is sometimes very intense, or the very close association, and they have the role to play. Each and every organism has a role to play in the life of the lichens. That is why now we call lichens as the consortium, or the lichens as the micro ecosystem. So therefore the definition of the lichens also changed since the period of Linnaeus. So Hawksworth, Hawksworth is a well-known lichenologist or a mycologist. See, he himself changed the definition of the lichens at least 8 times. So now the definition is that lichen is a self-sustaining ecosystem formed by the interaction of an exorbitant fungus and extracellular arrangement of one or more photosynthetic partners and an indeterminate number of microscopic organisms. So this is a definition of the lichens now. So then because of the association of the fungus and the algae in lichens, it gives several unique power or the unique ability to the lichens as a whole. So for example, lichens can grow on all kinds of substrata. For example, rock, moraine. Rock, rock is a very hostile substrate. There is no nutrition, but lichens can easily grow on rock. And the moraines, concrete, timber, old buildings, many things, even on the bones, boats, dead moss, glasses. So lichens can grow everywhere. And lichens grow on the most harsh climatic conditions like cold deserts or the hot deserts. Hot deserts like Thar, or the cold deserts like Antarctica or Leh Ladakh, lichens can easily grow there. But lichens are slow growing, their metabolic rate is very slow, and they are a very poor competitor. Unlike other organisms, unlike other fungus, lichen farming fungi are very poor competitors. So they are picolohydrous in the sense they don't have the control over their water content. If the water is available in plenty, they absorb the water. मैं एक लैपटॉप दे रहा हूँ, अभी मैं आता हूँ 11:30 बजे. So if the like the water is there, the lichens take up the water. If the water is not available, dry condition comes, they lose the water, and they become metabolically inactive. They remain physiologically inactive during the harsh condition, such as the cold conditions, extreme cold conditions, or low light, or even the harsh dry conditions. They remain physiologically inactive. When the favorable condition returns, they comes back to life. They are extremely tolerant to freezing stress, such as the temperature as low as -196 degree. They survive. They can survive. It has seen that in Antarctica they survive 96 weeks in the frozen condition at -15 degrees centigrade. For 10 years, under -20 degrees centigrade. In that cold condition, usually frost injuries are very common, but in the lichens, frost injuries is never seen. Like that, lichens can also photosynthesize in the low temperature as well as in the low light. So these are the unique abilities of the lichens. And as I told you, lichens they make very poor fossils. Their thallus, their body is very delicate. They don't form a good lichen thallus fossil. So that is why, although there are several fossils are discovered, several fossils are discovered since dating back to 2,800 years, but we could not interpret them as they are of lichens only, because they are very poorly preserved fossils. But the 415 million years ago, the fossils that were discovered, they are well preserved, and they showed all the structure of the lichens, the stratification, that upper layer, the algal layer, medulla, cortex, and all. It was clearly differentiated. So that is how we know that at least 415 million years back, lichens were evolved, lichens were formed. And what is the diversity of lichens? In the world, there are about 20,000 lichen species are there. People are giving different numbers at different times. And then some people said it is 13,000 to 13,500 to 22,000 and all. But we can take a middle number. We can say that 20,000 species of lichens are present. And the maximum diversity of lichens are present in the tropical areas of the world. And sometimes you get more number of lichens than the bird or the tree species you will get in that area. But unfortunately, these tropical areas are less exposed for the lichens. Because these tropical areas and the lichens, they grow in such a places which are inaccessible, and also they are much hostile. So the very inhospitable habitats there. So that is why lichens grow comfortably there, but we cannot access those areas for the exploration of the lichens. And also there are less number of people all over the world studying these lichens. Please mute your mic. So then coming to lichen research in India. So you know that before independence, the lichens of India were studied by the foreigners, especially the British, especially the Europeans. They collected the lichens. Some of them are mostly the army personnel. So doctors, they collected the lichens and took it back to Europe, and where they studied and published. And just before the independence, some people like Qureshi, Kashyap, and all, they started working on lichens. But their lichens, again, their collections were studied by the European lichenologist. Then the T.R. Seshadri, Biswas and all, they started working on the lichens of India. T.R. Seshadri, he was a chemist and who studied the chemistry of lichens, but credit goes to Biswas, who was a superintendent at Royal Botanical Garden, Calcutta. Now it is called as J.C. Bose Royal Botanical Garden or J.C. Bose Botanical Garden. So he studied the lichens of India, made a list of lichens, and he inspired Dr. D.D. Awasthi to do the lichen study. So then later on, D.D. Awasthi he took up the lichen study. He established the lichen center, research center at Lucknow University, and studied the lichens of the country. That is why Dr. Awasthi is called as the father of Indian lichenology. Then there are different people came. PG Patwardhan established the lichen center at this place at Maharashtra. Then Ajay Singh established the lichen center at Botanical Survey of India. Dr. Ajay Singh established the lichen research center at NBR. And then now at present, after Ajay Singh, Dr. Uprati studied the lichens at National Botanical Research Institute, and he diversified the area of research. Then came after the retirement of Dr. Uprati sir, now I am looking after the lichen division of the CSIR-NBR. So but the current condition is that in Lucknow University, lichen research has completely stopped, and Botanical Survey of India, we have only one researcher working on lichens. Even in the Agarkar Research Institute, there is only one person working on the lichen. So, but there are new centers are coming up. But in total, lichens people working on the lichens are reduced and not much. And this is the trend of lichen diversity. And we have a record of the lichen species added to the country, and the addition is going exponentially. And you can see the curve here, the graph here, it's going up and up. Currently, we have 3,900 taxa, 3,099 taxa, which makes 3,066 species, which comes under 4 subspecies, 28 varieties, and 1 pharma. And there are about 490 genera and 93 families are there. And it makes about 15% of the world known lichens. And then India, in India, there is an increase of about 543 species or percent, because if you compare it 1943, only 1947, only 477 species are reported. Now we have 3,066 species. It is a huge increase. That is 543% increase of lichens. You can see we ourselves keep discovering about 25 species, new species or new reports for the country. So there is a lot of scope for the lichen study in the country. And these are the diversity of lichens in the different regions. Now, for example, we make this country into 8 lichenogeographic regions. Among them, Western Himalaya makes the one of the biodiversity hotspots for lichens, which has 1,200 species, followed by Northeast India, including the Northeast Himalaya and all the states of Northeast India. Around 1,500 species are there. Then comes Andaman Nicobar Islands, where 525 species are there. Then Western Ghats, again rich in lichen diversity, where 1,472 species are there. And this is the diversity of lichens in different states. You can say Uttarakhand has the maximum diversity, 1,028 species. Uttarakhand has the maximum number because it is the one of the highly explored area. Because when the lichen research started in Lucknow University, and for the CSR NBRI also, Uttarakhand is the very accessible state, and both Dr. Awasthi and Dikyupratis are belong to Uttarakhand area. That is why exploration at Uttarakhand was easier, and also this Uttarakhand has the rich diversity. So that in total, that is why we have more number of lichens reported from the Uttarakhand. Then comes Tamil Nadu, then comes West Bengal. West Bengal is because although West Bengal is a plain area, it is mainly due to the Himalayan influence in the Darjeeling, then Arunachal Pradesh and all. If you see the right side of the graph, there are zero record of the lichens. It doesn't mean that there are no lichens. There are lichens, but people have not studied the lichens in the area. At least there are some lichens which are growing on the monuments in the dry conditions are available in this area. So if you study that area, at least some 25, 30 lichens, there is a possibility of getting. Then coming to the uses of the lichens, uses to human beings as well as uses in the ecosystem. Here you can see this is the best example we can give it for the lichens used in the ecosystem. This is the ecological succession. So ecological succession, where the one community of plants are replaced by the other community. So as I told you, lichens are the very sturdy organisms. They can easily grow on the rock. Although rock is a very hostile substratum, but lichens can easily grow there. And they in a barren rock in an area, lichens are the first organism to come and grow there. And once the lichens start growing there, they gradually make the place suitable for the other organism to come. They deteriorate the rock. They deteriorate the rock and make it soil, and also they accumulate soil also from the neighboring areas, and that is how they make the place suitable for the growth of the other organisms. Then comes the mosses or the bryophytes. Then the mosses and bryophytes are replaced by the grass and the weeds, and these grass and the weeds are then replaced by the herbaceous plants, and the shrubs will come, and like that one by one the community gets replaced, and one day you will see a climax forest area in that barren rock area. So this is a very slow process, but this is a very important process taking place in the nature in comparison to the when you see the geological scale. So the lichens help in the soil formation, that is called pedogenesis. It can be of both physical and the chemical process. In the physical process, what happens? The lichens growing on the rock, whenever the moisture is available, they absorb the moisture and they expand. And when the harsh condition comes during the you can say during the midday, so when the dry condition comes, they lose water, and then shrink. So they when the water available, they expand. This expansion and the shrinking of the lichen thallus creates pressure on the upper surface of the rock, and the soil particles gets detached from the surface. So then that particles becomes the soil particle. So that is how in the physical process it happens. At least this can happen thrice in a day. For example, in the morning and the evening time, there will be dew. So this the lichen thallus will expand. They take the moisture from the atmosphere and they expand. But during the midday, due to the hot sun, they lose the water and they shrink. Then comes the chemical process. These lichens which grow on the the rock surface, they secrete several chemical compounds. These chemical compounds interacts with the minerals of the rock, and the complex is formed, and the minerals or the upper surface of the rock get deteriorated. So this is called chelation process. So that is how the lichens growing on the rock surface, again through chemical process, they deteriorate the rock surface and may turn it into soil particles. So likewise, the lichens which are growing on the monuments, so they are very dangerous. That has to be removed. If you don't remove them, they deteriorate the rock surface. they give a shabby look to the rock surface. Some people say that lichens should be left to grow on the monuments because it gives an aesthetic sense and gives a sense that this is a very old monument. So for that you can keep, but if you want to preserve it, it is better you remove the lichens from the rock surface. Then the lichens are act as the food for the several of the microorganisms or the microinvertebrates. For example, nematodes, rotifers, oligochaeta, tardigrada. So, and n number of small insects, they eat these lichens as a food, and the snail also eats these lichens as a food. And this is called reindeer. Reindeer, the lichens is the favorite food of the reindeer. What it does, especially during the winter when there is no other plants available, grass and all is not available, these animals have the special sense for finding this, looking for this cladonia. This is a cladonia species you can see in the below picture. So they dig out the snow and they take out the lichen cladonia and they eat it. So that is why this cladonia is they are called as reindeer moss. And apart from that, these goats and all, they eat these lichens in the forest area. So sometimes these lichens are collected from the forest and fed to the animals in the animal farms. So the lichens is one of the major food of the deers. And apart from that, several small mammals, they eat these lichen. And one such mammal is northern flying squirrel, which has the very good link with the food chain and also bioaccumulation of the radio elements. So here the food chain, you can say these lichens are eaten by these northern flying squirrel, and these in northern flying squirrels are in turn eaten by the northern spotted owl. So, this during the Chernobyl accident took place in the Russia, where the nuclear reactor got leaked, and the nucleotides were radionucleic chemicals are they leaked into the atmosphere, and these were accumulated in the lichens. Okay, lichens are very sensitive to these pollutants and radioactive elements. Again, they were accumulated in the lichens. So these lichens are eaten by these reindeers, and these reindeers in turn they are eaten by the human beings. So the radioactive elements which are accumulated in the lichens, they directly went into the body of the human beings, which caused health issues. Then after the research, it is now declared that these deers are unsuitable for consumption. So this is one best example we can give for bioaccumulation as well as the food chain where lichens are involved. And this is a big list of animals or the small mammals which eat these lichens. And apart from that, lichens gives a shelter to the several of the microinvertebrates and the microbes. Protozoans, they make their the shelter on the lichen, like that mites, turgets and all. They also utilize these lichens for the shelter. And the lichens containing the cyanobacteria, the leptogium, peltigera and all, they fix the nitrogen. And once they fix the nitrogen, such lichens are, when they mix in the soil, they help in the increasing the fertility of the soil. And then also the other ecosystems are there. There are many ecosystems, services are there. Carbon fixing, the lichens help in the carbon fixing, especially in the polluted areas. Nutrients are recycling, denitrification, metal chelating, erosion prevention, water flow regulation, air quality regulation, plant germination and the growth support. Although these are the very, very small role played by the lichens, but they are important. And this is one of the area, the biological soil crust. So this you find in cold deserts in the arid zones. where lichens along with the other microorganisms, they make a crust, okay? And this crust is then will be present on the upper layer of the soil, and they have the lots of ecosystem service to provide. One important thing is that soil erosion. They in the arid region, either by wind or by snowfall, and which turns into water, so they lead to the soil erosion. But the soil biological soil crust, they prevent such soil erosion, and also these biological soil crust increase the fertility of the soil by adding the minerals and the nitrogen into the soil. So otherwise, these cold area, desert areas, or the arid zone, they're very harsh. So this improves the soil quality. So that is the another role of the biological soil crust and the lichens. And the lichens provide a beautiful background for the camouflage. You can, the first picture you can see there is a pepper moth which is sitting over the lichen thallus. Okay, so you cannot make out. That is how it is being protected from by their predator. Like that, in the below picture, you can see this is called catidid. Catidid insect. It looks like a grasshopper. It has taken the shape of the lichen thallus, lichen asthenia. You can see in the next to that insect is there. So that is how it is mimicking the lichen here. And here again, this insect is just looking like a lichen, and that is how it is being protected from the its predator. The one more insect is sitting over the leaf here, you can see. So it's just looking like a leaf. So these are the it's looking like a leaf, but on this leaf, the white spots you see, they're lichen. So the wings of the insect also taken the shape of the lichen. So apart from that, in this picture, you can see the frogs are sitting on the frogs are sitting over the lichen thallus, and they're nicely camouflaged by this. So that is how I mean they are camouflaged, and then they're safe from their predator. Okay, so again here you can see in this picture the this is one of the reptile lizard. It's a leaf-tailed gecko. It is growing. It is living. It is simply staying on the twig, and there you can clearly see the nicely it has been camouflaged here. Okay, 1 minute. So nicely it has been camouflaged here. So that is how lichens give a nice background. Here again you can see the this salamander. If it sits on the yellow lichen like this, you cannot make out. And this insect, it's a bag insect, bagworm. It is using the lichens for decorating itself, and also for living on the camouflaging on the background. So this is the hummingbird. You can see hummingbird using the lichens for the building its nest, and it's a small nest, but it's completely using the lichens for its nest building. And sometimes the birds use these lichens for the decoration purpose, but sometimes they use these lichens completely for the building the nest. You can see another bird is used they used lichens completely for the nest building. Here again you can see the lichens were laid on the these eggs were laid on the ground where the lichens were growing. So here the lichens, you can see the white thing is the lichens that makes a nice cushion here. So on that eggs are laid by the bird, and here the whole lichen thallus, the hanging one, is the lichens used by the birds for the building nest. So apart from that, how the lichens are useful to man, for useful to us, is that we people are daily using lichens in our food, but we don't know them. If you go to sometimes the markets and all, you will get the lichens in the supermarkets or Amazon's, Flipkarts and all. They sell the lichens as a spice. So in the name of stone flower. If sometimes in the big bazaar or Spencer and all, you will get lichens. So then lichens are used in the mute your mic, please. So some of the Maharashtrian masala, Goda masala, it's called Goda masala or Lavani masala, there the lichens are used. And Hyderabad biryani, lichens is used very much for the for flavoring. So lichens can be used as a raw, as a in Hindi we call as kada masala. You directly like you use this curry leaves like that you can use, but you can also make grind it, make it a powder, and also you can use it. So lichen acids, they don't give any smell, but when you put it when you fry a small piece of lichen in the ghee or the oil, it really releases the aroma. So that is how it gives a different flavor to the food. So and you know this person is called as Kunal Kapoor. So he's a good chef. So he is explaining in this video how the lichens are used, or the explaining about the lichens as a spice. So you can, link is given here in the YouTube. You can see this where he has clearly mentioning the uses of the lichens. And because of that, a large number of lichens are being collected from the Himalayas and from the Western Ghats, and then they are segregated, and only few species are used as the lichen spice. And then after segregation, they are sent into the different markets. So also they are exported outside the country. And that is why there is a different gradings of the lichens are there. So grade 1 lichen should have only one species, either Parmatrium nilgarensis or the Evernestum serratum. Only one species will be there. Grade 2 will have two species or so. Then grade 3 will have more than 3 species. So, but we collected lichens from the different markets and we studied them, and we found that at least there are 38 species of lichens are used as a spice, and the spice what you get in the market, it's a mixture of several species. So this is this is about the lichen spice. And lichens are also used for the perfumes. And again, I tell you here, lichens as such, they don't give you smell, but this is a process in which they give smell. So here, what we people do, the lichens are put into a big container along with the water and other herbs, and it is boiled. And the vapor that is generated from the after boiling this container, this lichen mixture, this vapor is passed through the small vials which is containing the essential oils. So the vapor, the lichen vapor passes through this essential oil, mixes it with that, and gives an entirely different smell. So the perfume which is prepared in such a way, they get sold for the highest prices. So this is how the lichens are used. And this is a picture of a place called as Kannauj. This place is in Uttar Pradesh. It's called as a perfume city because every household there, they make the perfume. What you can see here, the big containers are there. There the lichens and the other herbs are put, and you can see a pipe here coming out of it. And this at the end of this pipe, the container is kept, which is having the essential oils. And once these vapors go through this, and it mixes with that container, and it cools down and becomes liquid. And once it mixes with the essential oil, it gives a completely different smell. And lichens are also used in the dye. And so again here the lichens, although there are bright colors of lichens, again the process is very important. You know this litmus paper, which is used for the acid base indication. So these litmus papers are made up of lichens. So this is the lichen Roxella, is this lichen species which is used for the making the litmus paper. dye is extracted from this lichen, and this dye is impregnated over the paper, and then it is finally fixed, and that is how the lichen paper, litmus paper is made. And Switzerland is a country which dominates in making the litmus paper. So nowadays we have a pH meter, and the other method or the chemically prepared litmus papers are there. But earlier the litmus paper was prepared from the lichens. Here in this picture, you can see the wool and the tussar silk, cotton and all nicely can be boiled with the, nicely can be dyed. So this is the method. Either you can boil this lichen and the textile in the ammonia, or you can use the urea, or simply in the plain water. So it depends on the duration and the treatment method to give a different color. Here you can see it has given the nice golden yellow color here, the first picture. Here the lichens Lobelia pulmonaria is boiled with the wool lichens, and the this one it is being boiled. Similarly, like Parmatrima species has given after long time soaking, it has given a pretty violet color to the wool. Similarly, in the lower picture, Everest punastri was used here. Boiling water method is used. A little bit ammonia is added here. After soaking it for 10 days, it has given a beautiful yellow color. This is one experiment we made in our laboratory. We did both. We collected the lichens and the directly treated the tussar silk for the tussar silk for this coloring. You can see the beautiful color it has given. And also we cultured the microbiome part of these, some of these lichens, and it, tried it for dyeing, and it has given beautiful color. And also lichens are worn on the body during the rituals in the by the tribal people. So you can see in the first two picture, the lichen Asnia longissima is worn on the head and the face, and in the second picture, the mustache and the beard is made up of this lichen bryoria, the brown ones. So lichens were also used during the ethnic functions and ethnic events in the villages. So this is another the use of the lichens. If you are living in a cold temperate area, you can use this lichens for the aesthetics, for beautification. So you can make a nice artifacts or the potpourris using the lichens. You can see here how nicely these lichens were put in a container and made a memento, a piece of decoration. So you can make several artifacts also using these lichens. Then coming to the lichens as a medicinal use, and the lichens as the lichens having the medicinal importance goes back to long years. It may go up to 2,500 years or beyond that. And this is the mummy, female mummy called as Aptol, discovered from the Egypt pyramid. Then this lichen was taken to the Canada. I mean Harvard University. No, it was taken to Montreal Natural History Museum in Canada. There it was opened. Then they saw several herbs were there along with the mummy. It is a well-preserved mummy. And then they started interpreting why this herb is there and all. Among them, this major portion was of a lichen, Pseudoevernia porphyraceae. This lichen was sent to the Harvard University for identification. And they interpreted that why they have used a lot of lichens. One thing is that lichens being a soft thing, it keeps the mummy well preserved. Second thing is that lichens also has this aromatic property, this perfume giving property. So when the mummy is drying, it should not give the bad smell. That is why they have used these lichens. Or sometimes these lichens have lots of these medicinal properties. They have a lot of antimicrobial activities. So when the mummy is drying, and this it should not it should not decompose. it should dry, but it should not decompose. To avoid this microbial growth, these lichens were used. So these are the interpretation given for the usage of the lichens in the mummy. So then you can take any literature, the Hippocrates or the Theophrastus. So their literature gives the examples of the lichens used in the medicine throughout the world. And these are some of the common lichens mentioned in the all this literature for the medicinal purpose. Coming to India, I'm extremely sorry. So I'm coming to the India. So this lichens, reference of the lichens as they being used as the this one medicine. You can trace it back to Rigveda, Atharveda, or Sushruta Samhita, Charaka Samhita, and all. So all these literature, you will get the reference of the lichens being used in the medicine. And in Ayurveda, and the Nigantus also. In Ayurveda, the lichen is mentioned as the charila, and it is mainly refers to the lichens Parmelia perlata, which is now called as the Parmelia chinensis, and the large number of uses are mentioned there, like they are used as the astringent to resolve the resolvent, laxative, cumulative, and many things, even as the aphrodisiac. They are used in the spermatoria, amenorrhea, calculi, headache, bleeding. Many uses are mentioned in the Ayurveda. And also lichens are used in the tribal medicines. Tribes in the Sikkim, they are called Nepalese of Sikkim. They use this lichen heteroderma diadermata whenever bleeding is there, or the wound healing, as a wound healing. Like here, the Peltigera canina, this picture you can see. Here the lichens are used for the used by the lepchas for the stopping the bleeding. So also Chinese medicine lichen is one of the ingredient, and he is the Acharya Balakrishna of Patanjali. He has written a nice article, a big article. I have given the link here. He says that lichens can be used for the gray hair problem, headache, eye disease, urinary disease, erysipelas, wound healing, leprosy, itching problem, inflammation, and so many things. Yes, true, lichens can be used for all these purposes. And also the lichens, both their crude extract, lichen completely, the lichen thallus as such, or the lichen crude extract, and also isolated compounds from the lichens have shown lots of medicinal properties. So at least 150 lichens in India have shown the medicinal properties, biological activities. So among them, the antimicrobial studies are more common. At least 55 species have been utilized for the antimicrobial studies, and 57 lichen species are used for the antioxidant studies, and 37 species at least they are used for the anti-cancer studies. Some of the studies in progress are their hepatoprotective studies, cardiovascular protective, gastrointestinal problems, anti-obesity, anti-diabetic, probiotic, which are some of them are lifestyle disease, what we call. So Agarkar Research Institute, Swaminathan Institutes, Pune, Chennai, and CSIR-NBR are major contributor to the lichen prospection. So this is the list of lichen which are used mostly as a common lichen for the medicinal property studies or the bioprospection studies. And this is a big list of lichens which has shown biological activities against these many ailments or the problems. And why lichens are medicinally important? Because they produce more than 1,500 secondary metabolites. Among them, most of them are unique to lichens. You don't get them in anywhere else. So only 50-60 you may get in other organisms. So because these lichens have lots of secondary metabolites, these metabolites are responsible for the medicinal importance of the lichens. And then, as I told you, they have the therapeutic potential. but its therapeutical potentials are not yet explored fully. And arsenic acid is the widely medicinally used one of the metabolites, and this arsenic acid also utilized in the several product making. And you can see in this picture the algae or fungus are there. Algae produces these carbohydrates, and whereas the fungus produces the thallus formation and other things. But the series of set of experiment is too much here. The end product is completely different. So the lichens are also used for the nanoparticles preparation. It is called green chemistry. So silver nanoparticles and the gold nanoparticles are the two important nanoparticles which are synthesized by the using the lichen extracts. So the nanoparticles, they are the better agents than the using the direct medicines and all. They are environment friendly, cost effective, suitable for the pharmaceuticals and the biomedical applications. Many of these nanoparticles have shown the antimicrobial, antifungal, antiviral, and anti-pelletic activities, like the therapeutic agents, biomarkers, and the biodelivery vehicles. They act as biodelivery vehicles. So, like that, lichens are used also for the preparation of the these nanoparticles. So there is a n number of possibilities using the lichens for the bioprospection, because so lichens based commercially available products are very less. Arsenic acid is one of the compound, as I told you. Some of the antiseptic compounds are available in the market, especially in German. Again, this Italy, that one shaving cream was made out of lichens. And Icelandic moss or the Icelandic lichens are again called as they're used for the gold remedies formulation and Isamos institutes or so. So like that, there are few products available. These are the few products available for the lichens in the market, but not many. And but the problem is that lichens are very small organism. You don't get much myobasm or the bioprospection studies. What happens when you are into a bioprospection study? You need large amount of lichens or any plant material for the compound isolation and to make a product. But in case of lichens, they are already tiny organisms and they are also weighing very less, and it is difficult for getting the huge biomass. So that is why we have to look for the alternate method. Microbiome culture is one of the important method, because otherwise this lichen cannot be cultivated. Lichens cannot be grow in the market, grow in the garden. Lichens cannot be cultivated in the laboratory, cultured in the laboratory. That is why, because they don't survive, or even in the laboratory condition, if you grow them, they don't survive much. It will be a tedious job. That is why microbiome culture is possible. In the microbiome culture, what we do, we separate the fungus and the algae and then culture them separately using fungal media and the algal media. And there is a mixed media is there. And in the mixed media, we bring both fungus and the algae together, and then they co-cultivate them. So here again, the compatible algae and fungus should be there. It's a slow process, but the lichen thallus formation has been achieved in the recent days. So this is the protocol, or this is the flow diagrams where lichens are collected here, and either lichen thallus you can use, lichen apothecia. Here the lichen apothecia has been used, and pure form of lichens, lichen mycelia is available here in this picture. You can see. So here the pure thallus is mycelia is there, and the mycelia, these mycelia are made to produce the same secondary metabolites as there in the natural thallus. Although it is a difficult and tedious, but this can be achieved. Then this is how the method shown, one of the methods shown for the in vitro culture of the lichens here. Culture is again possible, but it is difficult in the laboratory. And full-fledged lichen thallus, you cannot get a full lichen thallus, even if it is developed, we cannot transplant them into the environment for the survival. It don't survive. These are the different microbiome thallus cultivation. You can see here are the lichen thallus culture. You can see how it is forming the lichen thallus here. So here what we do in the lichen culture, microbiome culture, this is the apothecia. The lower lid, you can see lower petri plates in the lower portion. The apothecia cut, apothecia thoroughly, this one sterilized with the running water and the other things. Then it is fixed onto the bottom petri plates using the glue, using the Vaseline or something like that, and the lid is put here. It's here actually the in the upper lid the media is there, and actually when you keep the this one apothecia here and keep it overnight for the sporulation, the spore actually breaks out of the ascus here and it jumps and it gets attached to the upper thallus, upper lid of the culture setup, petri plates. So then after a few days, you can take it out and you can see the mycelial growth here. So this will reduce the contamination. For this purpose, we have to wash it thoroughly, sterilize it, and use it on the, fix it on the lower lid of the thing, lid of the petri plates. So there are different medias are there for the cultivation of these microbiome. And then some of the research that has been carried out at CSIR-NBRI. For example, we have developed the nano antimicrobial microbials from the lichens. Fluconazole is a broad spectrum antifungal drug used for the treatment of the Candida albicans if the infection which at the high concentration rate. So that is fluconazole was isolated from the lichens, and the lichen dietary flavonoids quercetin isolated from the lichen Asnia longissima can be implemented as a sensitizing agent for the this chemical resistant this fluconazole, which is a broad spectrum antimicrobial. In that place you can use that. So several products we have developed, and this is one of the Swarna Bhasma is the Bhasma only, the ash only, that is prepared using the gold and the lichen extract. So this by addition of the lichen extract and the gold, it improves the quality of the ash. Even otherwise, this Bhasma after Havan and all, people use it for several purposes as a sacred thing. Even that has the medicinal properties. To enhance the medicinal property of that thing, Swarna Bhasma can be prepared. And the endolichenic fungi, I told you these lichens live in association with the other microorganisms. The fungus which is growing on the lichens are called as lichenicolous fungi, but inside the lichens are called as the endolichenic fungi. So, and in the endolichenic fungi, they're just like endophytes, and they also produce different unique metabolites, and this endolichenic fungi is again an interesting area for the biodiversity part, diversity part, as well as its medicinal properties. So lots of endolichenic fungi has been isolated, and they give a unique compounds and the very interesting biological activity reports. So hepatoprotective activity studies, as I've told you, it is going on in the sister laboratory, ERI Pune. And during the COVID-19 also, we tried that whether the lichen can be a useful organism for the extraction of the metabolites for during the COVID time. So it's chemicals which have been isolated from the lichens. They showed a very good antiviral activities against COVID also. And this is the, I'll not go deep into the paper, you can study this. This is that same thing. Another important role of lichens is in the air pollution indication. So they indicate the air pollution by bleaching and peeling off from the substratum. And if you have the record of the earlier lichens, we can clearly see the loss of diversity in the lichens and also disappearance of the sensitive species, you can clearly see. And also change in the lichen community structure, you can see. And apart from that, lichen also accumulate lots of heavy metals and the radioactive elements. So apart from that, lichens also indicate the mineral resource in a space, especially if a lichen is growing on a growing in a copper rich area, the lichens look bluish in color. And also by observing the nutrients from the substratum, sometimes these lichens also accumulate the minerals in that. And by looking it into the minerals, you can say that area is rich in what kind of minerals. And also lichens helps in the health of the indicates the health of the forest. If you, by looking at the community of the lichens, you can say about the health of the forest, whether it in a good condition or the bad condition or the deteriorated condition. Then comes the ecological continuity. You can say, looking at the lichen community, you can say that that is a continuous forest, or there was in between the disturbance were there like fire, and also the cutting is there, so it can indicate. So vegetation type, by looking at the simple lichen community, you can say what kind of vegetation is there, whether it's an evergreen forest or a deciduous forest, you can say. So apart from that, as I told you, the lichens are bio-indicators, bio-monitors, they indicate air pollution and all. So air pollution related studies were conducted since 1790, where Darwin observed that lichens failed to grow in a smelters, in the smelters. The lichens did not grow in Europe. So that is the first observation. Then by 1960, more than 200 research papers were published. Now we have more than 2,500 research papers published on the lichen research, lichen air pollution. And this is one of the examples we can say give where the lichens are used as a indicators of the air pollution, and very good correlation because based on the lichen population, the lichen diversity, and the people affected with the lung disease. This is the Veneto, a small town in the Italy. So what has happened? This is the same city here. The greener area in the first picture, you can see lots of diversity is there here. Wherever they place the green is there, lot of lichens are there. Lichen diversity is huge, and where the red portion is there, the lichen diversity is very less or no lichen diversity. So in the right picture, you can see the greener area corresponds to the lung cancer mortality. There is a lot of mortality due to the lung cancer. Where the bottom picture, you can see the red area. Sorry, I'll change it. Where the green portion is there is no mortality due to the lung cancer, or the less mortality is there. But the red portion, there is a lot of mortality is there because of the lung cancer. So the lung cancer is because of the air pollution. Whenever the air pollution is there, the lung cancer is there, the lichen diversity is not there. So wherever the lichens are there, there is no air pollution, and that area no death due to the lichen, the lung cancer. This is the close relation between the lichen diversity, air pollution. Like that, why lichens are very sensitive to air pollution? Because as I told you, 90% is of fungus and only 10% is algae is here. Algae is the very sensitive part. If the algae dies, whole lichen thallus dies. And algae has the chlorophyll in it. Chlorophyll, because of the air pollution, the iron content, iron molecules in the chlorophyll structure, if you see, it gets replaced by the magnesium, and like chlorophyll gets converted into pheophytin. The photosynthetic efficiency of the pheophytin is very less than the chlorophyll. So that is why the photosynthesis reduces. And lichens are directly depend on the atmosphere. Although some of the lichens have tiny root kind of structure, they are used for the anchoring purposes. Anchoring purpose, they don't really absorb the nutrients from the substratum. So that is why lichens are directly dependent on the atmosphere, and atmosphere is polluted, and this polluted directly go into the lichen thallus. And lichens do not have any protective layer, cuticle layer. So any protection is not there. Lichen thallus is very sponge-like, hygroscopic. So that is why the pollutant directly go into the lichen thallus. And their lichens are much active during the wet conditions. So, and also that is why wet conditions, the pollutants are much accumulated in the lichen thallus. And then there is no shedding structures, leaf sheddings, or leaf shedding structures is not there. So throughout the lifetime, lichens keep on accumulating these heavy metals and the pollutant. And lichens are perennial. So the sensitive species, they die. Sensitive species are the folios and the fruticose lichens. They're sensitive, they die. But the tolerant ones, they remain. So the tolerant ones, they tolerated the pollution and they survive. Such lichens are used for the lichen pollutant accumulation studies. So lichens are cosmopolitan in distribution. They have a wide range of geographical distributions, and they are perennial organism. They're available throughout the area. They are cheap and the cost effective. So that is why the lichens are used for the biomonitoring studies. So using lichens is in the air pollution studies much cheaper than using the equipments. So this is how the lichens in sulfur dioxide affect the lichens. This is what's shown here. So the chlorophyll, finally, because of the presence of the sulfuric acid, chlorophyll gets broken into pheophytin, and the magnesium is released here. And this magnesium, chlorophyll is iron is replaced by the magnesium. That is what is shown here. And the lot of indication of the sulfur dioxide injury into the lichens, bleaching, reduction in the thallus size, reduction in the cell organelles, many things are there here. And this is well studied. Again, the microbiome, changes in the microbiome and the photobiome because of the pollution, how the alter structure is changed here, that is mentioned here. And as I told you, fruticose lichens and the foliose lichens are much sensitive than the crustose lichens. Crustose lichens is much tolerant here. And we can study the lichens. You can study the air pollution using the lichens here. One thing is zone mapping. We call. We can make a different zones out of lichen presence here. Distribution we can make a distribution map of a particular species. You can see its diversity, density here. So usually near the air pollution source, the diversity will be less, and the accumulation of the lichen pollutant in the lichen will be less. There is something called as index of atmospheric purity. Lichen diversity value index is there, and we can make the present and lichen diversity with the past lichen diversity studies. We can make and draw the conclusion. We can also make active. By naturally growing lichens, we can do several studies. If the lichens are not growing in an area, we can transplant the lichens from the healthier area to a polluted area and keep it there for 1 month or so. And then you bring it to the laboratory and study. You can see the tremendous accumulation of the pollutants in these lichens. So that is called lichen transplantation study. That is the active bioindicator studies. The passive is the one where you are using the naturally occurring lichens. And accumulator bioindicator studies means it is accumulates the pollutants. That is what they study. And reactive indicators means some of the lichen response biological lichens physiology changes in response to the air pollution. So that is called reactive bioindicator studies. So here you can see the zone mapping here. The center portion is the lichen desert. There are no lichens because it is directly exposed to the pollution. There is a pollution source here. But if you move on the 4 directions, you will initially get inner struggle zone. Inner struggle zone, you will get only few crustose lichens which are tolerant to pollutions. You will get. Then you will get outer struggle zone. There you will get tolerant crustose lichen. Some foliose lichen we will get here. And then the transition zone where you will get normal foliose lichens will start growing here. The outer zone where there is no pollution, the normal lichens you can get here. So then this is a study which we carried out for the Uttar Pradesh, Lucknow here. The city center, you can say there are no lichens here. Lichen desert is there here. And if you go a little away from the area, city centers are known for the traffic activity. If you go away from the city center, you will get the normal lichen growth. This is the air pollution study we did for the Unchahar. So as we go away from the this nuclear thermal power plant, the lichen diversity increased and also density increased. This is what we want to tell in this paper. And index of the atmospheric purity is by this formula we can know it gives us some value out of 10. 1 to this out of 10 it gives you some value, and larger the value of IAP, cleaner is the air. This is the conclusion we draw. So larger the value, the better is the air quality. That is the conclusion we draw here. And this is the zone mapping. We can make a different zones out of the using the lichen diversity here again. And this is one of the study which I carried out for the Karnataka, Bangalore. Here we emphasized on the Lalbagh. Here we can see that in 1980s, already one studies had been carried out by the Upreti and Avasti. And I repeated the study for the Lalbagh garden in 2003. In 20 years of gap, there is a lot of change. The foliose lichens diversity has come down drastically, whereas the crustose lichen diversity has increased. So foliose lichens being the sensitive, their number reduced because of the urbanization and the pollution in the neighboring area. So similarly, in the crustose lichens also they reduced in number, or the crustose lichen increased. You can clearly say in 1988 numbers were there. In 2003, there are 12 species were there because crustose lichens were the pollution tolerant ones. There are only few common species between the two. Only 4 common species of the foliose lichens. You can see here. So otherwise, the crustose lichens, there are no common lichens. The completely community structure change is there in here in this area. Similarly, Kolkata study, Kolkata also we studied. For the botanical garden in Howrah, in 1867, Nylander has already studied, and after 200 years, we studied the area. Upreti et al. We studied. We can see the drastic change in the lichen community and then the diversity.
Sir,
so 53. Yes.
Sir, time is up actually. So how much time?
Yeah, I'll conclude.
Okay, okay, sir. Yeah, a resource person is waiting. So, sir, sorry.
So I'm told that I'm given 1.5 hours, up to 11:30. But if the time is up, I have no problem. I'll
okay, thank you, sir.
So this is what the different use of the lichens. This is why I was telling the lichen transplant study, where the lichens are from the healthy areas are brought to the polluted area, and it's kept for 1 month there, and the pollutant accumulated by them are studied. So see again the polycyclic aromatic hydrocarbons. They are again the pollutants in the air. The lichens can also indicate the presence of polycyclic aromatic hydrocarbons in the lichens. So these are the other parameters. This is that comparison. Which one is cheaper using the equipment or the lichens? This is what I'm trying to show here. Nowadays, the nitrogen pollution is the very big problem because we use a lot of nitrogen fertilizers and the nitrogen fertilizers, which finally leaches into the soil and the atmosphere and create a lot of problem. And lichens are being used for the studying this one. This is what I'm trying to show. And the lichens also indicate the climate change. So we are also using the lichens for the studying the climate change. And this is also one of the study called the lichenometry, where the lichens are used for the studying the glacier movement caused by the climate change. This is the different setups for the studying the climate change. We are continuously studying the climate change by setting up a permanent research plots in the Jammu Kashmir, Himachal Pradesh, Uttarakhand, Sikkim, and the Arunachal Pradesh. These are the permanent plots that we have established in these areas, hilly areas. These are directly monitored by the satellite, and online data is being collected. And conservation of the lichens, because for the spice purpose and other things, large number of lichens are being collected, as I told you earlier. And there is no protocol has been so far developed for the conservation of the lichens. And none of the lichens of the Indias are recorded in the Red Data book. And although we have proposed some of the areas, lichen-rich areas, as the lichen sanctuary to be proposed, and in the future, at least some of them will be result in the lichen sanctuaries. And some of the beautiful lichens like these can be introduced in the lichen garden for the conservation purpose. And one such lichen garden is already come up in the Munsheri areas of the Uttarakhand. So one conservatory has come up in the Malabar Botanical Research Garden, Malabar Botanical Research Institute, where the lichens are lichen conservatory has been introduced. Swaminathan gave the first lichen for the conservation purpose. So this is it. Thank you. If you have any questions, and if there is a provision for the provision for the question and answer, you can ask me. Okay. Thank you very much for the patience here. Yes.